Lab 3 is on microscopes, and below is an example of a microscope. So let's label some of the parts here. Let's use red. So the eyepiece here, we can call these oculars. Ocular, and there's two of them. These guys are adjustable, and some of them might contain a pointer, which is useful in pointing things out. Next we have is the arm of the microscope, right here. And down here at the base, we aptly call this the base of the microscope. Whenever you carry a microscope, one hand on the arm and the other under the base. Okay, this rotating piece right here, these are the objectives. So here is the yellow objective, here is the blue objective. Below it, where you place the slide, we call this the stage. So this is the stage. Right here, this is your power switch. And then this is one of the light controls. Okay. These two knobs right here, these are the stage controls. They move the stage away from you and also left and right. So they control the stage. We call them stage controls. Stage controls. Okay. So same microscope, different perspective. Again, these are your objectives. These are your objectives. Again, this here is your stage. And these clips that secure the stage, we call them stage clips. So clips right there. Okay, so these two knobs right here, they bring the stage up and down closer to the objectives. This tool here, we call this the coarse adjustment. And this up here, this is the fine adjustment. So you first start off using the coarse, but once you can start to see something, use the fine to focus what you're staring at. And then lastly, this here is the light source. Light source. Okay, so before you use your microscope, just make sure to clean the objectives and oculars with lens paper. All right, now how to create a slide. So let's kind of draw out what we are starting with. You usually have some kind of container with some kind of liquid. Say this is water for now. You have your specimen. Let's say it's an Elodea leaf in this case, so it's a leaf. The slide glass, a rectangular piece of glass. And then in a cover slip box, you will find cover slips. And these are very easily breakable, so be careful. And then we have tweezers. So step one is to apply 2 to 3 drops at the center. So step one would be to apply 2 drops in the center. Okay, step 2, with forceps, you are going to place your Elodea leaf on the center where you just placed that water. So here are my forceps placing that leaf. So step 2 is place the specimen, doesn't have to be a leaf, but place specimen. Place specimen in the center. with the forceps. Okay, so step 3 is with the forceps, you are going to, there is my water, here is my leaf. With the forceps, you're going to take the cover slip and you're going to place it on one side. So here's my cover slip. Haven't dropped it yet, it's just placed on one side with my forceps. So step 3 is with forceps. You're going to place the cover slip on one side. On one side. And then you're going to gently lower it to cover the other side. So it will go down this way. And then place gently, or I guess you can release it to the other side. Then place gently on other side. So in the last step, you will have something that looks like this. So there is my, my specimen, and the water that was just there kind of got squished, so it distributed itself, it got compressed. And then here is my cover slip. So now what you're going to do is with a pen or a pencil, you're just going to press on this, on the cover slip carefully to remove any air bubbles. So with a pen or a pencil, you are going to press gently on the cover slip. And this is going to remove your air bubbles. But that's it. So the first thing we're going to be looking at are our cheek cells, but it might be hard to see because they're transparent. To help us see things, we stain cells. So that is why we stain things to see the cell and its components. So things that are inside the cell and its components. Okay. So like I said, the first thing we'll be looking at are our cheek cells, and let me kind of give you an idea of what that looks like. So circular-ish, not perfect circle. On the inside, you'll see little specks, distributed. And then approximately in the center, we will have our nucleus. So let's label this structure. So this covering is called the cell membrane. Kind of any region inside the cell, like here for example, we call this cytoplasm. And in this case, we can see these little specks, these granules. We'll call this a granular cytoplasm, just to be a bit more specific. Granular cytoplasm. Okay. And then lastly, this structure in the center, this is our nucleus, which contains a few important things. Mainly contains your DNA. Your nucleus is composed of a nuclear membrane. And it also has a darker region in the center. That darker dot you see in the center, this is called the nucleolus. And we'll speak about that in a sec. Okay, so let me take my nucleus. So I'm going to take my nucleus and kind of blow it up to get an idea of what we're looking at. Let's use purple. So here is my nucleus. Again, this is the nuclear membrane, just like any cell has a membrane, so is a nucleus. And along this membrane, we'll have these little pores. these openings. So these are called nuclear pores. And this is how things enter and exit the nucleus. Inside, we'll see this loose, this is actually DNA in its loose configuration called chromatin. So again, nucleus contains DNA. This is the DNA right here. But in this loose form, we call it chromatin. It's still DNA though. DNA in chromatin form. Okay, and lastly, we have a darker dot in the center. This guy here, this is called a nucleolus. Nucleolus. And let's talk about this for a sec. This contains ribonucleic acid, or we can abbreviate this to RNA. And ribonucleic acid is used to produce ribosomes, to make ribosomes. So we see ribonucleic acid, ribo makes ribosomes. And then ribosomes actually make proteins. So ribosomes make proteins. So RNA makes ribosomes, ribosomes makes proteins. Okay. And last thing I want to mention is that the ribosomes aren't in the nucleus, they actually leave. So the ribosomes, they exit the nucleus and they reside just outside it in something called the endoplasmic reticulum. Reside in, its full name is rough endoplasmic reticulum. That's where it does its job of making protein. So we abbreviate this to rough RER. Okay, so here is your cheek cell. Not much to see here. We'll just be focusing on cytoplasm versus nucleus versus the cell membrane. Okay, so I will see you in the next video.